Once again, I am Mark Giulianotti, and I'm a Program Director at the Center for the Advancement of Science in Space, or CASIS. It's my pleasure to be presenting this webinar covering a new ISS National Lab research announcement. And as we get started with this presentation, I just wanted to remind everyone that this webinar is being recorded and will be published on our website. If you have any questions, please enter them in the questions dialog box. We will have some time at the end of this webinar to address some of the questions. If we do not get to your question today, we will post a full list of questions and answers when we put the video on our website. So with that, let's get started. This is our 6th National Lab research announcement of 2021. You can find more information regarding the previous 5 on our website. This 6th one is focused in the area of in-space production applications, and specifically in the areas of tissue engineering and biomanufacturing. As I mentioned in my introduction, I work for CASIS, and CASIS is responsible for managing the ISS National Lab on the ISS. And we do this in partnership with NASA. At the National Lab, we have access to up to 50% of the resources available to the US on the ISS. This includes astronaut time, facilities use, space and launch vehicles, as well as the return vehicles. We utilize this resource to drive scientific breakthroughs, advance commercial space opportunities, and inspire the next generation. We do that with a focus on driving the benefits for those of us here on Earth. Where NASA has a focus on utilizing the ISS, in part to test technologies that will reduce risks for space exploration, our focus at the National Lab is on returning tangible benefits to those of us here on Earth. So please keep that key point in mind when responding to the solicitation. The primary reasons to do work in space are to take advantage of these key attributes: the extreme environment, the vantage point, and the sustained microgravity. You can utilize the outside of the platform to expose materials to the vacuum of space, extreme temperature variations, radiation, as well as microparticle impacts. As the station orbits the Earth, it provides a vantage point to 90% of the world's populated areas, and is the only human-tended satellite, thus enabling the crew the ability to real-time modify when and where they record images. However, most germane for this announcement is the microgravity, which alters many observable phenomena within the physical and life sciences. Moreover, microgravity induces a vast array of changes in organisms, ranging from viruses and bacteria to humans, including global alterations in gene expression and three-dimensional aggregation of cells into tissue-like architecture. So that brings us to the specifics of this announcement. The keys are, this is for in-space production applications, and specifically in the areas of tissue engineering and biomanufacturing. So I'll spend some time providing some key background on what these mean in the context of this solicitation. But I'll also encourage you to read the full announcement and also reach out to us if you have questions about whether your concept is in scope for the solicitation. So in-space production. First, this is not an opportunity focused on fundamental research questions. We have other opportunities to support those research goals. As I mentioned earlier, part of our role at the National Lab is to contribute to the commercial space economy, utilizing the ISS as a testbed for commercial opportunities. This announcement is really focused on that aspect. The proposals must discuss how the project could grow new business and capital investment, and ultimately return long-term value for continued access to the space environment. The proposal should specify how they would utilize the ISS through this opportunity to mature their process and/or validate their production capabilities. The proposals must focus on how the LEO environment uniquely enables the specific project proposed. So you must address why you can't do this here on Earth. The ultimate goal for this program is that projects will be matured and validated so that they will be ready for scale-up as we see more and more commercial space platforms become available. Tissue engineering and biomanufacturing covers areas that utilize biological materials that are engineered to be used for clinical applications themselves or as tools for the discovery of novel therapeutics. It could range from using single-cell populations to multicellular applications, and in fact could include non-cellular biological products. But again, if you have questions as to whether your concept is appropriate for this solicitation, please reach out. And once again, as a reminder, if you have any questions throughout this webinar, please put them in the question dialog box. With that initial introduction, let me give you an overview on why we are interested in supporting this opportunity. As many of you know, we have seen amazing technological advancements in recent years in the areas of biological research, such as gene editing and the use of iPSCs, to name just a couple. We've also seen a tremendous increase in the integration of engineering and miniaturization into research platforms, as well as massive leaps forward in analytical capabilities. I think everyone is convinced that tissue engineering will provide substantial beneficial impacts to the health and well-being of our citizenship. However, in the field, we are still confronted by a number of challenges. And while we see hope in the number of clinical trials ongoing, we have yet to see the full expanse of significant impacts. While there are a number of factors that contribute to that, we are hopeful that the use of the ISS could help expedite some of these future uses. Again, while we all agree the tools and technologies available to us today are significantly advanced from those just a decade or two ago, the time and cost for novel therapeutic development continues on average to increase. So the question is, can a LEO-based platform be utilized to open up new opportunities not available terrestrially, and will these confer a commercially sustainable advantage? So for example, stem cells and stem cell-derived products hold potential as both research tools and therapeutic products. One of the earliest initiatives that we undertook at the National Lab was to support stem cell research on the ISS. Working off some preliminary indications that spaceflight has an effect on stem cells, we asked the research community, is there an advantage to culturing and expanding stem cells in space? Or what knowledge could be applied to terrestrial applications based on doing this research in space? This was work that was fully supported by CASIS in hopes that it would help develop more advanced cell culture techniques, reveal scientific breakthroughs, and lead to more initiatives. Some of this work has led to the validation of several key findings, such as clinically relevant stem cells can be expanded on the ISS, and the expansion in LEO appears to confer some improvements in their capabilities over those expanded on Earth. Also, that culturing in microgravity can reveal novel targets to go after in order to optimize cellular products that are designed for transplantation applications. Additionally, real-time imaging and live return of cell cultures from space is possible. And we've seen an increase in LEO cell culture capabilities from our community of implementation partners. So for this current announcement, you could potentially highlight ways to gain insights into how to control and optimize stem cell pluripotency and multipotency, proliferation, expansion, differentiation, and/or maturation of stem cells and stem cell derivatives through the use of a LEO-based platform, and how this could lead to commercial investment in this LEO-based application. This would be an example of being in scope for this solicitation. There's an intense interest in aging research, which aims to prolong our healthy and productive years. Novel approaches to enhancing quality of life include targeting cellular senescence, genomic instability, and telomerase shortening. It is known that spaceflight induces a number of physiological changes in both astronauts and rodent models. Skeletal muscle atrophy, bone density loss, immune dysfunction, cardiovascular deconditioning, and arterial stiffening, for example. These changes resemble aging-related maladies on Earth and are well documented in extremely healthy astronauts. These findings provide a compelling case for studying astronaut health by researchers seeking to understand disease processes, especially because these changes in spaceflight occur very fast compared to their ground-based analogs, and they are reversible upon return to Earth. They also open up an opportunity to utilize multicellular devices, such as tissue chips, to model these fast onsetting disease phenotypes that can rapidly return to baseline. Some initial work in collaboration with the NIH, specifically the National Center for Advancing Translational Sciences, NCATS, has enabled significant advances in the hardware capabilities to utilize these microphysiological systems on the ISS. These systems have capabilities ranging from partially to fully automated and have been developed in partnership with the implementation partner community. Scientifically, some key initial new findings include identifying a novel link between immune system and stem cell senescence, 2 different teams showing spaceflight cardiac dysfunction in 3D models, and some new omics level details on how disuse attributes to post-traumatic osteoarthritis. These studies are still ongoing, as well as others supported by NCATS and NSF. Now, the opportunity under this announcement would be to identify how these tissue chip platforms or other biologically engineered systems could be utilized in LEO to drive sustained revenue, perhaps as a platform for novel target identification or validation studies, or as preclinical safety and efficacy testing platforms. Similarly, we at the National Lab have supported several organoid payloads. These self-organized 3D tissue cultures could be integrated into tissue chip platforms, or they could be independent of the engineered platforms and potentially enable higher throughput screening capabilities, or even be brought down for use terrestrially after being cultured in LEO and distributed as commercial products. Biofabrication covers a wide section of opportunities, ranging from fabricating tissues for disease modeling, testing of biofabricated materials, and improving fabrication processes for those biomaterials. As noted earlier, there's still an urgent need for tissues, organs, and other biomaterials for use in transplantations and regenerative medicine applications. In the past decades, many novel tools have emerged to enable the assembly of these complex structures. All of these terrestrial-based tools and techniques integrate or attempt to circumvent the effects of gravity in their process. Given that there are still wide needs for additional biofabrication techniques, there are potential advantages to biofabricating in space. These include the use of less viscous bioinks, ability to enhance cell-cell interaction, such as for organoid production, ability to print simultaneously from any spatial orientation, the ability to control mechanotransduction effects due to gravity during the maturation and curing process. There are currently commercially operated biofabrication systems on the ISS, including the TechShot biofabrication facility shown here. Again, proposals that look to build commercial opportunities in this area are within scope of this announcement. To repeat, this solicitation is really focused on the commercial opportunities within the tissue engineering and biomanufacturing fields. We will be looking to support projects that identify some of these key points. The direct link to the solicitation is here, and in a minute I will go over some of the details regarding the application process, as well as the timeline and context. Those details can also be found at the website. In the solicitation documents, you will see that we have identified some priority areas, also listed here. These are the same areas that I briefly covered earlier. We are open to other areas outside these identified, but if you have any questions regarding your specific concept, please feel free to reach out to us to discuss them. One area, such as novel drug formulations or development of polymorphs or other crystalline technologies, including those involving proteins, is likely going to be more appropriate for our currently open NLRA number 5. Again, if you have questions, please reach out to us. A research announcement opened on March 22nd and follows a 2-step proposal process. Step 1 is the concept summary. The concept summary is a 2 to 3 page summary of your proposal. Concept summaries must be received no later than May 6th. The concept summary will be evaluated internally for operational feasibility and scientific merit. We'll then invite those summaries that pass review acceptance to submit a full proposal in Step 2. Step 2 is the full proposal and will be by invitation only. It will be due June 22nd. If selected, we will award a fully funded total scope mission, including launch, payload integration, in-orbit mission costs, data return, and if applicable, payload sample return. No funding will be granted to cover the offeror's internal project costs. As noted, the first step is the concept summary, 2 to 3 pages in length. There is a template provided, and please use this template. You will see that we are asking for some of the basics on what your hypothesis is, what metrics you will utilize to measure success, why the ISS is necessary, and how your experiment will work operationally. As noted previously, if you have any questions on these items, please reach out to us. Ken Saven is the lead contact for this, and his email is listed here. Ken is also joining me on this webinar for the Q&A session. For the operational approach, I have noted throughout this webinar the implementation partner community, who have developed hardware to support research in this area and have the expertise to implement your ISS experiments. On the solicitation, we provide a list of hardware available and implementation partners, but we can also help to introduce you to these companies, so please reach out to us at ops@issnationallab.org if you need help in identifying an implementation partner to work with. For the compliance, we are requesting that the PI and co-PIs must be U.S. persons and that your institute or company be a U.S. company or institute. In the solicitation documents, you will find a rubric of how the proposal will be evaluated. However, at the high level, they will be evaluated based on operational feasibility, scientific merit, and benefit to life here on Earth. Please reach out to our operations team with any questions about available hardware and working with the implementation partners. As far as funding, we anticipate providing approximately $1.5 million in funding for the solicitation, with the expectation of making up to 2 awards. Again, the funding will go to support all the mission integration and operations costs. No funding will be granted to cover the offeror's internal project costs. From a contractual standpoint, it is anticipated that CASIS will execute a subcontract directly to the implementation partner, so the awarded PI's institute or company will not need to do this. Once again, I would like to encourage you to contact us if you have any questions. I thank you for your time and wish you the best of luck in your submission. We are really excited about this opportunity and the future of InSpace production. And with that, we'll begin our question and answer period. So if you haven't already, please enter your questions in the question dialog box.